Insect morphology. Insect morphology is the study and description of the physical form of insects. The terminology used to describe insects is similar to that used for other arthropods due to their shared evolutionary history. Separate insects from other arthropods. They have a body divided into three regions, called tagmata, head, thorax, and abdomen, three pairs of legs, and mouthparts located outside of the head capsule. The position of the mouthparts divides them from their closest relatives, the non-insect hexapods, which include Protura, Dipleura, and Collembola. Insect morphology. Legend of body parts. Tagmata: A. Head, B. Thorax, C. Abdomen. 1. Antenna. 2. Ocelli, lower. 3. Ocelli, upper. 4. Compound eye. 5. Brain, cerebral ganglia. 6. Prothorax. 7. Dorsal blood vessel. 8. Tracheal tubes, trunk with spiracle. 9. Mesothorax. 10. Metathorax. 11. Forewing. 12. Hindwing. 13. Midgut, stomach. 14. Dorsal tube, heart. 15. Ovary. 16. Hindgut, intestine, rectum and anus. 17. Anus. 18. Oviduct. 19. Nerve cord, abdominal ganglia. 20. Malpighian tubes. 21. Tarsal pads. 22. Claws. 23. Tarsus. 24. Tibia. 25. Femur. 26. Trochanter. 27. Foregut crop, gizzard. 28. Thoracic ganglion. 29. Coxa. 30. Salivary gland. 31. Subesophageal ganglion. 32. Mouthparts. There is enormous variation in body structure amongst insect species. Individuals can range from 0.3 mm fairy flies to 30 cm across. Great owlet moth 1-7 have no eyes or many, well-developed wings or none, and legs modified for running, jumping, swimming, or even digging. These modifications allow insects to occupy almost every ecological niche except the deep ocean. This article describes the basic insect body and some variations of the different body parts. In the process, it defines many of the technical terms used to describe insect bodies. Anatomy summary. Insect arthropods have no interior skeleton. Instead, they have an exoskeleton, a hard outer layer made mostly of chitin that protects and supports the body. The insect body is divided into three parts: the head, thorax, and abdomen. Used for sensory input and food intake. The thorax, which is the anchor point for the legs and wings, if present, is specialized for locomotion, and the abdomen is for digestion, respiration, excretion, and reproduction. 1.22.8 Although the general function of the three body regions is the same across all insect species, there are major differences in basic structure, with wings, legs, antennae, and mouthparts being variable from group to group. 3. External. Exoskeleton. The skeleton, the cuticle, consists of two layers: the epicuticle, which is a thin, waxy, water-resistant outer layer that lacks chitin, and the layer under it is called the procuticle. This is chitinous and much thicker than the epicuticle and has two layers. The outer is the exocuticle while the inner is the endocuticle. The tough and flexible endocuticle is built from numerous layers of fibrous chitin and proteins, crisscrossing each other in a sandwich pattern, while the exocuticle is rigid and sclerotized. 22-24 The exocuticle is greatly reduced in many soft-bodied insects, especially the larval stages, e.g., caterpillars. Chemically, chitin is a long-chain polymer of an acetylglucosamine, a derivative of glucose. In its unmodified form, chitin is translucent, pliable, and resilient. In arthropods, however, it is often modified, becoming embedded in a hardened proteinaceous matrix, which forms much of the exoskeleton. In its pure form, it is leathery, but when encrusted in calcium carbonate, it becomes much harder. The difference between the unmodified and modified forms is evident when comparing the body wall of a caterpillar unmodified to a beetle modified. From embryonic stages, a layer of columnar or cuboidal epithelial cells gives rise to the external cuticle and an internal basement membrane. The majority of insect material is inside of the endocuticle. The cuticle provides muscular support and acts as a protective shield as the insect develops. Since it cannot grow, the external sclerotized part of the cuticle is periodically shed in a process called molting. As the time for molting approaches, most of the exocuticle material is reabsorbed. In molting, the old cuticle separates from the epidermis apolysis. Enzymatic molting fluid is then released between the old cuticle and epidermis, which separates the exocuticle by digesting the endocuticle and sequestering its material for the new cuticle. When cuticle has formed sufficiently, the epicuticle and reduced exocuticle are shed in. Ecdysis. 5:16-20. The formable regions of an insect body segment are the tergum or dorsal, sternum or ventral, and the two pleura or laterals. Hardened plate exoskeleton are called sclerites, which are subdivisions of the major regions, tergites, sternites, and pleurites, for respective regions tergum, sternum, and pleuron. 6. Head. Head Orthoptera, acrididae. A. Antenna. B. C. Vertex. D. Compound I. E. Occiput. F. Genna. G. Fleurostoma. H. Mandible. I. Labial palp. J. Maxillary palps. K. Maxilla. L. Labrum. M. Clypeus. N. Frons. Larva beetle, family Cerambicidae, showing sclerotized epicranium, rest of body hardly sclerotized. Larva of syrphid fly, member of Cycloropha, without epicranium, almost without sclerotization apart from its jaws. The head in most insects is enclosed in a hard, heavily sclerotized, exoskeletal head capsule. Makes in those species whose larvae are not fully sclerotized, mainly some Holometabola, but even most unsclerotized or weakly sclerotized larvae tend to have well sclerotized head capsules, for example, the larvae of Coleoptera and Hymenoptera. The larvae of Cycloropha, however, tend to have hardly any head capsule at all. The head capsule bears most of the sensory organs, including the antennae, ocelli, and compound eyes, along with the mouthparts. In the ovate, the head capsule appears unsegmented, though embryological studies show it to consist of 6 segments that bear the paired head appendages, including the mouthparts, each pair on a specific segment. 7. Each such pair occupies one segment, though not all segments in modern insects bear any visible appendages. All the insect orders, Orthoptera displays the greatest variety of features found in the heads of insects, including the sutures and sclerites. Here, the vertex, or the apex dorsal region, is situated between the compound eyes of insects with hypognathous and opistognathous heads. In proboscis insects, the vertex is not found between the compound eyes, but rather where the ocelli are normally found. This because the primary axis of the head is rotated 90 degrees to become parallel to the primary axis of the body. In some species, this region is modified and assumes a different name. 8.13. The extra is made of the coronal, frontal, and epicranial sutures plus the ecdysial and cleavage lines, which vary among different species of insects. The ecdysial suture is longitudinally placed on the vertex, separating the epicranial halves of the head to the left and right sides. In insect, the suture may come in different shapes, like either A, Y, U or V. Those diverging lines that make up the ecdysial suture are called the frontal or frontogenal sutures. Species of insects have frontal sutures, but in those that do, the sutures split open during ecdysis, which provides an opening for the new instar to emerge from the integument. The frons is that part of the head capsule that lies ventrad or anterior of the vertex. The frons varies in size relative to the insect, and in many species, the definition of its borders is arbitrary, even in some insect taxa that have well-defined head capsules. In species, though, the frons is bordered at its anterior by the frontoclypeal or epistomal sulcus above the clypeus. Laterally it is limited by the frontogenal sulcus, if present, and the boundary with the vertex, by the ecdysial cleavage line, if it is visible. If there is a meanocellus, it generally is on the frons, though in some insects such as many Hymenoptera, all 3 ocelli appear on the vertex. A more definition is that it is the sclerite from which the pharyngeal dilator muscles arise, but in many contexts that too, is not helpful. In the anatomy of some taxa, such as many Cicadomorpha, the front of the head is fairly clearly distinguished and tends to be broad and subvertical, that median area commonly is taken to be the frons. 9. The clypeus is a sclerite between the face and labrum, which is dorsally separated from the frons by the frontoclypeal suture in primitive insects. The clypeus is a sclerite between the face and labrum, which is dorsally separated from the frons by the frontoclypeal suture in primitive insects. The clipogenal suture laterally demarcates the clypeus, with the clypeus ventrally separated from the labrum by the clipolabral suture. Clypeus differs in shape and size, such as species of Lepidoptera with a large clypeus with elongated mouthparts. The cheek or genna forms the sclerotized area on each side of the head below the compound eyes extending to the gular suture. Like many parts making up the insect's head, the genna varies among species, with its boundaries difficult to establish. In dragonflies and damselflies, it is between the compound eyes, clypeus, and mouthparts. The genna is the area immediately posterior, or posterior or lower on the genna of pterygote insects, and forms the lateral and ventral parts of the occipital arch. The occipital arch is a narrow band forming the posterior edge of the head capsule arching dorsally over the foramen. The area is usually narrow, located above the mouthparts. This area also includes the hypostoma and fluorostoma. 8-13 The vertex extends anteriorly above the bases of the antennae as a prominent, pointed, concave rostrum. The posterior wall of the head capsule is penetrated by a large aperture, the foramen. It passes the organ systems, such as the nerve cord, esophagus, salivary ducts, and musculature, connecting the head with the thorax. 10. The aspect of the head or the occiput, postgena, occipital foramen, posterior tentorial pit, gula, postgenal bridge, hypostomal suture and bridge, and the mandibles, labium, and maxilla. The occipital suture is well founded in species of Orthoptera, but not so much in other orders. Where found, the occipital suture is the arched, horseshoe-shaped groove on the back of the head ending at the posterior of each mandible. The occipital suture is a landmark on the posterior surface of the head, and is typically near the occipital foramen. In goats, the postocciput forms the extreme posterior, often U-shaped, which forms the rim of the head extending to the postoccipital suture. In PT or goats, such as those of Orthoptera, the occipital foramen and the mouth are not separated. The three occipital closures, or points under the occipital foramen that separate the two lower halves of the postgena, are the hypostomal bridge, the postgenal bridge, and the gula. The hypostomal bridge is usually found in insects with hypognathous orientation. The postgena is found in the adults of species of higher Diptera and aculate Hymenoptera, while the gula is found on some Coleoptera, Neuroptera, and Isoptera, which typically display prognathous-oriented mouthparts. 8-15. Compound eyes in ocelli. Most have one pair of large, prominent compound eyes composed of units called ommatidium, singular up to 30,000 in a single compound eye of, for example, large dragonflies. This takes resolution than eyes found in vertebrates, but it gives an acute perception of movement and usually possesses UV and green sensitivity, and may have additional sensitivity peaks in other regions of the visual spectrum. Often an ability to detect the vector of polarized light exists in polarization of light. 11. There can also be an additional 2 or 3 ocelli, which help detect low light or small changes in light intensity. The input is a combination of inputs from the numerous ommatidia, located on a convex surface, thus pointing in slightly different directions. Compared with simple eyes, compound eyes possess very large view angles and better acuity than the insect's dorsal ocelli, but some stemmatal equals larval eyes, for example, those of sophli larva tenthredinity with an acuity of 4 degrees and very high polarization sensitivity, match the performance of compound eyes. 12. 12-13. Ocellus cross-section. Compound eye cross-section. Individual lenses are so small, the effects of diffraction impose a limit on the possible resolution that can be obtained, assuming they do not function as phased arrays. This can only be countered by increasing lens size and number. To see the resolution comparable to our simple eyes, humans would require compound eyes that would each reach the size of their heads. Compound eyes fall into two groups: apposition eyes, which form multiple inverted images, and superposition eyes, which form a single erect image. 14-15. Compound eyes grow at their margins with the addition of new ommatidia. 16. Antennae. Close-up of a fire ant, showing fine sensory hairs on antennae. Antennae, sometimes called feelers, are flexible appendages located on the insect's head which are used for sensing the environment. Insects can feel with their antennae because of the fine hairs, setae, that cover them. 17. 8-11. However, touch is not the only thing that antennae can detect. Numerous tiny sensory structures on the antennae allow insects to sense smells, temperature, humidity, pressure, and even potentially sense themselves in space. 17. 8-11. 18. 19. Some insects, including bees and some groups of flies, can also detect sound with their antennae. The number of segments in an antenna varies amongst insects, with higher flies having 3 to 6 segments, 21, while adult cockroaches can have over 140. 20. The general shape of the antennae is also quite variable, but the first segment, the one attached to the head, is always called the scape, and the second segment is called the pedicel. The remaining antennal segments or flagellomers are called the flagellum. 17. 8-11. General insect antenna types are shown below. Types of insect antennae. 1. 17. 8-11. Aristate, capitate, clavate, filiform, flabellate, geniculate, setaceous lamellate, maniliform, pectinate, plumose, serrate, stellate. Mouthparts. The insect mouthparts consist of the maxilla, labium, and in some species, the mandibles. 8-16-23. The labrum is a simple, fused sclerite, often called the upper lip, and moves longitudinally. It is hinged to the clypeus. The mandible's jaws are a highly sclerotized pair of structures that move at right angles to the body, used for biting, chewing, and severing food. The maxillae are paired structures that can also move at right angles to the body and possess segmented palps. The labium, lower lip, is the fused structure that moves longitudinally and has a pair of segmented palps. 24. Insect mouthparts. From the primitive chewing mouthparts of a grasshopper in the center A, to the lapping type B, of a bee, the siphoning type C, of a butterfly, and the sucking type D, of a female mosquito. Legend. A. Antennae. C. Compound eye. LB. Labium. LR. Labrum. MD. Mandibles. MX. Maxillae. The mouthparts and rest of the head can be articulated in at least 3 different positions: prognathous, opistognathous, and hypognathous. In species articulation, the head is vertically aligned with the body, such as species of Formicidae, while in a hypognathous type, the head is aligned horizontally adjacent to the body. An opistognathous head is positioned diagonally, such as in species of Blatodia and some Coleoptera. 25. The mouthparts vary greatly between insects of different orders, but the 2 main functional groups are mandibulate and haustellate. Stellate mouthparts are used for sucking liquids and can be further classified by the presence of stylates, which include piercing sucking, sponging, and siphoning. The stylates are needle-like projections used to penetrate plant and animal tissues. The stylates and the feeding tube form the modified mandibles, maxilla, and hypopharynx. 24. Mandibulate mouthparts, among the most common in insects, are used for biting and grinding solid foods. Piercing sucking mouthparts have stylates and are used to penetrate solid tissue and then suck up liquid food. Sponging mouthparts are used to sponge and suck liquids, and lack stylates, e.g. most diptera. Siphoning mouthparts lack stylates and are used to suck liquids and are commonly found among species of Lepidoptera. Many mouthparts are found in species of Odonata, adult Neuroptera, Coleoptera, Hymenoptera, Blatodia, Orthoptera, and Lepidoptera. However, most adult Lepidoptera have siphoning mouthparts, while their larvae, commonly called caterpillars, have mandibles. Mandibulate. The labrum is a broad lobe forming the roof of the preoral cavity, suspended from the clypeus in front of the mouth and forming the upper lip. 122 to 24 on its inner side. It is membranous and may be produced into a median lobe, the epipharynx, bearing some cincilla. The labrum is raised away from the mandibles by two muscles arising in the head and inserted medially into the anterior margin of the labrum. It is closed to the mandibles apart by two muscles arising in the head and inserted on the posterior lateral margins on two small sclerites, the tormae, and, at least in some insects, by a resolute spring in the cuticle at the junction of the labrum with the clypeus. 26. Until recently, the labrum generally was considered to be associated with the first head segment. However, recent studies of the embryology, gene expression, and nerve supply to the labrum show it is innervated by the tritocerebrum of the brain, which is the fused ganglia of the third head segment. This is formed from the fusion of parts of a pair of ancestral appendages found on the third head segment, showing their relationship. 22 to 24. Its ventral, or inner, surface is usually membranous and forms the lobe-like epipharynx, which bears mechanosencilla and chemisencilla. 27-28. Chewing insects have two mandibles, one on each side of the head. The mandibles are positioned between the labrum and maxillae. The mandibles cut and crush food, and may be used for defense. Generally, they have an apical cutting edge, and the more basal molar area grinds the food. They can be extremely around 3 on moss, or an indentation hardness of about 30 kg per square millimeter. Thus, many termites and beetles have no physical difficulty in boring through foils made from such common metals as copper, lead, tin, and zinc. 122 to 24. The cutting edges are typically strengthened by the addition of zinc, manganese, or rarely, iron, in amounts up to about 4% of the dry weight. 7. They are typically the largest mouthparts of chewing insects, being used to masticate, cut, tear, crush, chew food items. They open outwards to the sides of the head and come together medially. In chewing insects, the mandibles can be modified to be more knife-like, whereas in herbivorous chewing insects, they are more typically broad and flat on their opposing faces, e.g., caterpillars. In stag beetles, the mandibles are modified to such an extent as to not serve any feeding function but are instead used to defend mating sites from other males. In ants, the mandibles also serve a defensive function, particularly in soldier casts. In bull ants, the mandibles are elongated and toothed, used as hunting and defensive appendages. Situated beneath the mandibles, paired maxillae manipulate food during mastication. Maxillae can have hairs and teeth along their inner margins. At the outer margin, the galea is a cupped or scoop-like structure, which sits over the outer edge of the labium. They also have palps, which are used to sense the characteristics of potential foods. They occupy a lateral position, one on each side of the head behind the mandibles. The part of the maxilla consists of a basal cardo, which has a single articulation with the head, and a flat plate, the stipes, hinged to the cardo. Both cardo and stipes are loosely joined to the head by a membrane, so they are capable of movement. Distally on the stipes are two lobes, an inner lacinia, and an outer galea, one or both of which may be absent. More laterally on the stipes is a jointed, leg-like palp made up of many segments. In Orthoptera, there are 5. Anterior and posterior rotator muscles are inserted on the cardo, and ventral adductor muscles arising on the tentorium are inserted on both the cardo and stipes. Stipes are flexor muscles of the lacinia and galea, and another lacinial flexor arises in the cranium, but neither the lacinia nor the galea has an extensor muscle. The palp has levator and depressor muscles arising in the stipes, and each segment of the palp has a single muscle causing flexion of the next segment. 26. Ungulate mouthparts. The labium is a quadrupedal structure, although it is formed from two fused secondary maxillae. It can be described as the floor of the mouth. With the mouth, it assists with the manipulation of food during mastication or chewing, or, in the unusual case of the dragonfly nymph, extends out to snatch prey back to the head, where the mandibles can eat it. The labia is similar in structure to the maxilla, but with the appendages of the two sides fused by the midline, so they come to form a median plate. The basal part of the labium, equivalent to the maxillary cardines and possibly including a part of the sternum of the labial segment, is called the postmentum. This may be subdivided into a proximal submentum and a distal mentum. Distal to the postmentum, and equivalent to the fused maxillary stipets, is the prementum. The prementum closes the preoral cavity from behind. Terminally, it bears 4 lobes, 2 inner glossae, and 2 outer paraglossae, which are collectively known as the ligula. One or both pairs of lobes may be absent or they may be fused to form a single median process. A palp arises from each side of the prementum, often being 3-segmented. 26. The hypopharynx is immediately behind the mouth, projecting forwards from the back of the preoral cavity. It is a lobe of uncertain origin, but perhaps associated with the mandibular segment. 26 In aptery goats, earwigs, and nymphal mayflies, the hypopharynx bears a pair of lateral lobes, the supralinguae, singular, supralinguae. It divides the cavity into a dorsal food pouch, or sabarium, and a ventral salivarium into which the salivary duct opens. 122-24 It is commonly found fused to the labium. Most of the hypopharynx is membranous, but the odoral face is sclerotized distally, and proximally contains a pair of suspensory sclerites extending upwards to end in the lateral wall of the stomodium. Muscles arising on the frons are inserted into these sclerites, which distally are hinged to a pair of lingual sclerites. These, in turn, have inserted into them antagonistic pairs of muscles arising on the tentorium and labium. Various muscles serve to swing the hypopharynx forwards and back, and in the cockroach, two more muscles run across the hypopharynx and dilate the salivary orifice and expand the salivarium. 26 Diversity of mandibles. Piercing sucking. Mouth parts can have multiple functions. Some combine piercing parts along with sponging ones which are then used to pierce through tissues of plants and animals. Female mosquitoes feed on blood, hemiphagus, making them disease vectors. Mosquito mouthparts consist of the proboscis, paired mandibles and maxillae. The maxillae form needle-like structures, called stylets, which are enclosed by the labium. When mosquito bites, maxillae penetrate the skin and anchor the mouthparts, thus allowing other parts to be inserted. The sheath-like labium slides back, and the remaining mouthparts pass through its tip and into the tissue. Then, through the hypopharynx, the mosquito injects saliva, which contains anticoagulants to stop the blood from clotting. And finally, the labrum upper lip is used to suck up the blood. Species of the genus Anopheles are characterized by their long palpi, two parts with widening end, almost reaching the end of labrum. 29. Diversity of piercing mouthparts. Mosquito. Cymex lectularius. Flea. Horsefly, female. Tick, Ixodes ricinus, an arachnid for comparison. Siphoning. The proboscis is formed from maxillary galleae and is an adaptation found in some insects for sucking. 30. The muscles of the sabarium or pharynx are strongly developed and form the pump. In Hemiptera, which feed on fluids within plants or animals, some components of the mouthparts are modified for piercing, and the elongated structures are called stylets. Combined tubular structures are referred to as the proboscis, although specialized terminology is used in some groups. Species of Lepidoptera, it consists of two tubes held together by hooks and separable for cleaning. Each tube is inwardly concave, thus forming a central tube through which moisture is sucked. Suction is effected by the contraction and expansion of a sac in the head. 31. The proboscis is coiled under the head when the insect is at rest and is extended only when feeding. 30. The maxillary palpi are reduced or even vestigial. 32. They are conspicuous and five-segmented in some of the more basal families and are often folded. 8. And dimensions of the proboscis have evolved to give different species wider and therefore more advantageous diets. An allometric scaling relationship between the body mass of Lepidoptera and length of the proboscis. 33, from which an interesting adaptive departure is the unusually long-tongued hawk moth Xanthopon morgani praedicta. Charles Darwin predicted the existence and proboscis length of this moth before its discovery based on his knowledge of the long-spurred Madagascan star orchid Angraecum sesquipedale. 34. Siphoning mouthparts. Sponging. Insects that feed on fluids are modified in various ways to form a tube through which liquid can be drawn into the mouth and usually another through which saliva passes. The muscles of the sabarium or pharynx are strongly developed to form a pump. In non-biting flies, the mandibles are absent and other structures are reduced, the labial palps have become modified to form the labellum, and the maxillary palps are present, although sometimes short. In Brachycera, the labellum is especially prominent and used for sponging liquid or semi-liquid food. The labella are a complex structure consisting of many grooves, called pseudotracheae, which sop up liquids. Salivary secretions from the labella assist in dissolving and collecting food particles so they can be more easily taken up by the pseudotracheae or laid their egg on the suitable media. This is thought to occur by capillary action. The liquid food is then drawn up from the pseudotracheae through the food channel into the esophagus. 36. The mouthparts of bees are of a chewing and lapping sucking type. Mode of feeding in which liquid or semi-liquid food adhering to a protrusible organ, or tongue, is transferred from substrate to mouth. Honeybee Hymenoptera, Apidae, Apis mellifera. The elongated and fused labial glossae form a hairy tongue, which is surrounded by the maxillary galleae and the labial palps to form a tubular proboscis containing a food canal. the tongue is dipped into the nectar or honey, which adheres to the hairs, and then is retracted so the adhering liquid is carried into the space between the galleae and labial palps. This back and forth glossal movement occurs repeatedly. Movement to the mouth results from the action of the sebareal pump, facilitated by each retraction of the tongue pushing liquid up the food canal either for feeding requirements or to have a suitable media for laying their egg. 1:22-24. Sponging mouthparts. Thorax. The thorax has three segments: the prothorax, mesothorax, and metathorax. Anterior segment, closest to the head, is the prothorax. Its major features are the first pair of legs and the pronotum. The middle segment is the mesothorax. Its major features are the second pair of legs and the anterior wings, if any. The third, the posterior, thoracic segment, abutting the abdomen, is the metathorax, which bears the third pair of legs and the posterior wings. Each segment is delineated by an intersegmental suture. Each segment has four basic regions. The dorsal surface is called the tergum or notum, to distinguish it from the abdominal terga. 22-24. The two lateral regions are called the pleura, singular, pleuron, and the ventral aspect is called the sternum. The notum of the prothorax is called the pronotum, the notum for the mesothorax is called the mesonotum, and the notum for the metathorax is called the metanotum. Continuing with this logic, there are also the mesopelora and metapelora, as well as the mesosternum and metasternum. 8. Tergal plates of the thorax are simple structures in apterygotes and many immature insects but are variously modified in winged adults. Thoracic nota each have two main divisions: the anterior, wing-bearing alinotum, and the posterior, phragma-bearing postnotum. Phragmata, singular. Phragma are plate-like apodums that extend inwards below the antecostal sutures, marking the primary intersegmental folds between segments. Phragmata provide attachment for the longitudinal flight muscles. Each alinotum, sometimes confusingly referred to as a notum, may be traversed by sutures that mark the position of internal strengthening ridges and commonly divide the plate into three areas: the anterior prescutum, the scutum, and the smaller posterior scutellum. The lateral pleural sclerites are believed to be derived from the subcoxal segment of the ancestral insect leg. Sclerites may be separate, as in silverfish, or fused into an almost continuous sclerotic area, as in most winged insects. 1:22-24. Prothorax. The pronotum of the thorax may be simple in structure and small in comparison with the other nota, but in beetles, mantids, many bugs, and some orthoptera, the pronotum is expanded, and in cockroaches, it forms a shield that covers part of the head and mesothorax. 1:22-24. Pterothorax. The mesothorax and metathorax hold the wings. They have a combined name called the pterothorax. Pteron equals wing. The forewing goes by different names in different orders, e.g., the tegnina in Orthoptera and elytra in Coleoptera arises between the mesonotum and the mesopeloron, and the hindwing articulates between the metanotum and metapeloron. The legs arise from the mesopeloron and metapelora. The mesothorax and metathorax each have a pleural suture, mesopleural and metapleural sutures, that runs from the wing base to the coxa of the leg. The sclerite anterior to the pleural suture is called the episternum, serially, the mesepisternum and metepisternum. The sclerite posterior to the suture is called the epimyron, serially, the mesepimyron and metepimyron. Spirits, the external organs of the respiratory system, are found on the pterothorax, usually one between the pro and mesopleoron, as well as one between the meso and metapleoron. 8. The sternum follows the same convention, with the prosternum under the prothorax, the mesosternum under the mesothorax and the metasternum under the metathorax. The pleura and sternum of each segment have a variety of different sclerites and sutures, varying greatly from order to order, and they will not be discussed in detail in this section. 8. Wings. Most phylogenetically advanced insects have two pairs of wings located on the second and third thoracic segments. 1:22-24. Insects are the only invertebrates to have developed flight capability, and this has played an important part in their success. Insect flight is not very well understood, relying on turbulent aerodynamic effects. The primitive insect groups use muscles that act directly on the wing structure. More advanced groups making up the Neoptera have foldable wings, and their muscles act on the thorax wall and power the wings indirectly. 24. These muscles can contract multiple times for each single nerve impulse, allowing the wings to beat faster than would ordinarily be possible. Insect flight can be rapid, maneuverable, and versatile, possibly due to the changing shape, extraordinary control, and variable motion of the insect wing. Insect orders use different flight mechanisms. For example, the flight of a butterfly can be explained using steady state, non-transitory aerodynamics, and thin airfoil theory. Cross-section of wing vein. Internal. Each of the wings consists of a thin membrane supported by a system of veins. Formed by two layers of integument closely opposed, while the veins are formed where the two layers remain separate and the cuticle may be thicker and more heavily sclerotized. Within each of the major veins is a nerve and a trachea, and, since the cavities of the veins are connected with the hemocoel, hemolymph can flow into the wings. 6. As the wing develops, the dorsal and ventral integumental layers become closely opposed over most of their area, forming the wing membrane. The remaining areas form channels, the future veins, in which the nerves and tracheae may occur. The cuticle surrounding the veins becomes thickened and more heavily sclerotized to provide strength and rigidity to the wing. Hairs may occur on the wings, microtrichia, which are small and irregularly scattered, and macrotrichia, which are larger, socketed, and may be restricted to veins. The scales of Lepidoptera and Trichoptera are highly modified macrotrichia. 27. Veins. Venation of insect wings, based on the Comstock-Needham system. In some minuscule insects, the venation may be reduced. In chalcidoid wasps, for instance, only the subcosta and part of the radius are present. An increase in venation may occur by the branching of existing veins to produce accessory veins or by the development of additional, intercalary veins between the original ones, as in the wings of Orthoptera, grasshoppers and crickets. Large numbers of veins are present in some insects, and they may form a reticulum as in the wings of Odonata, dragonflies and damselflies, and at the base of the forewings of Tedigonioidea and Acridoidea, katydids and grasshoppers, respectively. 26. The arctectian is the name given to a hypothetical scheme of wing venation proposed for the very first winged insect. It is based on a combination of speculation and fossil data. Since all winged insects are believed to have evolved from a common ancestor, the arctectian represents the template that has been modified and streamlined by natural selection for 200 million years. According to current dogma, the arctectian contains 6 to 8 longitudinal veins. Veins and their branches are named according to a system devised by John Comstock and George Needham, the Comstock-Needham system. 30. These veins and their branches are named according to a system devised by John Comstock and George Needham, the Comstock-Needham system. 37. Costa C, the leading edge of the wing. Subcosta S, longitudinal vein behind the costa, typically unbranched radius R, third longitudinal vein. 1 to 5 branches reach the wing margin media M, fourth longitudinal vein. 1 to 4 branches reach the wing margin cubitus Cu, fifth longitudinal vein. 1 to 3 branches reach the wing margin anal veins A1, A2, A3, unbranched veins behind the cubitus. The costa C is the leading marginal vein on most insects, although a small vein, the precosta, is sometimes found above the costa. In all extant insects, 141 to 42, the precosta is fused with the costa. The costa rarely ever branches because it is at the leading edge, which is associated at its base with the humeral plate. The trachea of the costal vein is perhaps a branch of the subcostal trachea. Located after the costa is the third vein, the subcosta, which branches into 2 separate veins, the anterior and posterior. The base of the subcosta is associated with the distal end of the neck of the first axillary. The fourth vein is the radius, which is branched into 5 separate veins. The radius is generally the strongest vein of the wing. Toward the middle of the wing, it forks into a first undivided branch R1 and a second branch, called the radial sector Ra, which subdivides dichotomously into 4 distal branches R2, R3, R4, R5. Basally, the radius is flexibly united with the anterior end of the second axillary 2-ax. 38. The fifth vein of the wing is the media. In the R-type pattern, the media forks into 2 main branches, a media anterior MA, which divides into 2 distal branches MA1, MA2, and a median sector, or media posterior MP, which has 4 terminal branches M1, M2, M3, M4. In modern insects, the media anterior has been lost, and the usual media is the 4-branched media posterior with the common basal stem. In the Ephemer, according to present interpretations of the wing venation, both branches of the media are retained, while in Odonata, the persisting media is the primitive anterior branch. The media is often united with the radius, but when it occurs as a distinct vein, its base is associated with the distal median plate M, or is continuously sclerotized with the latter. The cubitus, the sixth vein of the wing, is primarily 2-branched. The primary forking takes place near the base of the wing, forming the 2 principal branches Cu1, Cu2. The anterior branch may break up into several secondary branches, but commonly it forks into two distal branches. Branch of the cubitus Cu2 in Hymenoptera, Trichoptera, and Lepidoptera was mistaken by Comstock and Needham for the first anal. Proximally, the main stem of the cubitus is associated with the distal median plate M of the wing base. 38. The postcubitus PCU is the first anal of the Comstock and Needham system. The postcubitus, however, has the status of an independent wing vein and should be recognized as such. In nymphal wings, its trachea arises between the cubital trachea and the group of vanal tracheae. In the more generalized insects, the postcubitus is always associated proximally with the cubitus and is never intimately connected with the flexor sclerite 3-ax of the wing base. In Neptera, Mecoptera, and Trichoptera, the postcubitus may be more closely associated with the vanal veins, but its base is always free from the latter. The postcubitus is usually unbranched. Primitively, it is two-branched. The vanal vein to NV are the anal veins immediately associated with the third axillary, and are directly affected by the movement of this sclerite that brings about the flexion of the wings. In number, the vanal veins vary from 1 to 12, according to the expansion of the vanal area of the wing. Vanal tracheae usually arise from a common tracheal stem in nymphal insects, and the veins are regarded as branches of a single anal vein. Distally, the vanal veins are either simple or branched. The jugal vein J of the jugal lobe of the wing is often occupied by a network of irregular veins, or it may be entirely membranous. Sometimes it contains one or two distinct, small veins, the first jugal vein, or vena arcuata, and the second jugal vein, or vena cardinalis 2J. 38. CSC cross veins, run between the costa and subcosta. R cross veins, run between adjacent branches of the radius. RM cross veins, run between the radius and media. MCU cross veins, run between the media and cubitus. All the veins of the wing are subject to secondary forking and union by cross veins. In some of insects, the cross veins are so numerous, the whole venational pattern becomes a close network of branching veins and cross veins. Ordinarily, however, a definite number of cross veins having specific locations occurs. The more constant cross vein H between the costa and subcosta, the radial cross vein R between R and the first fork of Rs, the sectorial cross vein S between the two forks of R8, the median cross vein MM between M2 and M3, and the mediocubital cross vein MCU between the media and the cubitus. 38. The insect wings are characterized by a convex-concave placement, such as those seen in mayflies, i.e., concavas, down, and convexas, up, which alternate regularly, and by their branching, whenever a vein forks there is always an interpolated vein of the opposite position between the two branches. The concave vein will fork into two concave veins, with the interpolated vein being convex, and the regular alteration of the veins is preserved. 30. The veins of the wing appear to fall into an undulating pattern according to whether they tend to fold up or down when the wing is relaxed. The basal shafts of the veins are convex, but each vein forks distally into an anterior convex branch and a posterior concave branch. Thus, the costa and subcosta are convex and concave branches of a primary first vein. Rs is the concave branch of the radius, posterior media is the concave branch of the media, Cu1 and Cu2 are respectively convex and concave, while the primitive postcubitus and the first vanal have each an anterior convex branch and a posterior concave branch. The concave nature of the veins has been used as evidence in determining the identities of the persisting distal branches of the veins of modern insects, but it has not been demonstrated to be consistent for all wings. 2638. Fields. Areas are delimited and subdivided by fold lines, along which the wings can fold, and flexion lines, which flex during flight. Between the flexion and the fold lines, the fundamental distinction is often blurred, as fold lines may permit some flexibility or vice versa. Two concepts found in nearly all insect wings are the claval, a flexion line, and jugal folds, or fold line, forming variable and unsatisfactory boundaries. Wing folding can be very complicated, with transverse folding occurring. Insect morphology. Antennae, SG. Antennae, sometimes referred to as feelers, are paired appendages used for sensing in arthropods. Large antennae on a longhorn beetle. Antennae are connected to the first one or two segments of the arthropod head. They vary widely in form but are always made of one or more jointed segments. While they are typically sensory organs, the exact nature of what they sense and how they sense it is not the same in all groups. Functions variously include sensing touch, air motion, heat, vibration, sound, and especially smell or taste. Antennae are sometimes modified for other purposes, such as mating, brooding, swimming, and even anchoring the arthropod to a substrate. 2. Larval arthropods have antennae that differ from those of the adult. Many crustaceans, for example, have free-swimming larvae that use their antennae for swimming. Antennae can also locate other group members if the insect lives in a group, like the ant. The common ancestor of all arthropods likely had one pair of uniramous, unbranched, antenna-like structures, followed by one or more pairs of biramous, having two major branches, leg-like structures, as seen in some modern crustaceans and fossil trilobites. Except for the chelicerates and proterans, which have none, all non-crustacean arthropods have a single pair of antennae. 4. Crustaceans. Cutaway diagram of a barnacle, with antennae highlighted by arrow. Crustaceans bear two pairs of antennae. The pair attached to the first segment of the head are called primary antennae or antennules. This pair is generally uniramous, but is biramous in crabs and lobsters and remipedes. The pair attached to the second segment are called secondary antennae or simply antennae. The second antennae are plesiomorphically biramous, but many species later evolved uniramous pairs. Second antennae may be significantly reduced, e.g. remipedes, or apparently absent, e.g. barnacles. Divisions of crustacean antennae have many names, including flagellomers, a shared term with insects, annuli, articles, and segments. The terminal ends of crustacean antennae have two major categorizations, segmented and flagellate. An antenna is considered segmented if each of the annuli is separate from those around it and has individual muscle attachments. Flagellate, on the other hand, have muscle attachments only around the base, acting as a hinge for the flagellum, a flexible string of annuli with no muscle attachment. 5. There are several notable non-sensory uses of antennae in crustaceans. Many crustaceans have a mobile larval stage called anopleus, which is characterized by its use of antennae for swimming. Barnacles, a highly modified crustacean, use their antennae to attach to rocks and other surfaces. 2. Antennae in the burrowing Hippoidea and Choristidae have setae that interlock to form a tube or snorkel, which funnels filtered water over the gills. 6. A spiny lobster, showing the enlarged second antennae. The large flattened plates in front of the eyes of a slipper lobster are the modified second antennae. A spiny lobster. Crustacean. A spiny lobster, showing the enlarged second antennae. The large flattened plates in front of the eyes of a slipper lobster are the modified second antennae. Crab Cancer pygurus, showing its reduced antennae. Antennules of the Caribbean hermit crab. Anopleus larva of a shrimp with antennae. Used for swimming. Examples of crustacean antennae. Copepoda, isopoda, amphipoda, decapita, remipedia, cirripedia. M. Aryantanae. Secondar. Insects. Terms used to describe shapes of insect antennae. Antennal shape in the Lepidoptera from C. T. Bingham, 1905. Some claim insects evolved from prehistoric crustaceans, and they have secondary antennae like crustaceans, but not primary antennae. Antennae are the primary olfactory sensors of insects, 7, and are accordingly well equipped with a wide variety of cincilla, singular, cincillum. Paired, mobile, and segmented, they are located between the eyes on the forehead. Embryologically, they represent the appendages of the second head segment. 8. All insects have antennae, however they may be greatly reduced in the larval forms. Amongst the non-insect classes of the hexapoda, both Collembola and Dipleura have antennae, but Protura do not. 9. Antennal fibrillae play an important role in Culex pipiens mating practices. The erection of these fibrillae is considered to be the first stage in reproduction. These fibrillae serve different functions across the sexes. Antennal fibrillae are used by female C. pipiens to locate hosts to feed on. Male C. pipiens utilize them to locate female mates. Structure. Electron micrograph of antenna surface detail of a wasp, Vespula vulgaris. The three segments of the typical insect antenna are the scape or scapus base, the pedicel or pedicellus stem, and finally the flagellum, which often comprises many units known as flagellomers. 10. The pedicel, the second segment, contains the Johnston's organ, which is a collection of sensory cells. 11. The scape is mounted in a socket in a more or less ring-shaped sclerotized region called the torulus, often a raised portion of the insect's head capsule. The socket is closed off by the membrane into which the base of the scape is set. However, the antenna does not hang free on the membrane, but pivots on a rigidly sprung projection from the rim of the torulus. That projection on which the antenna pivots is called the antennifer. The whole structure enables the insect to move the antenna as a whole by applying internal muscles connected to the scape. The pedicel is flexibly connected to the distal end of the scape, and its movements in turn can be controlled by muscular connections between the scape and pedicel. The number of flagellomers can vary greatly between insect species, and often is of diagnostic importance. Flagellomers are connected by membranous linkage that permits movement, though the flagellum of true insects does not have any intrinsic muscles. Some other arthropoda do, however, have intrinsic muscles throughout the flagellum. Such groups include the Symphyla, Collembola and Dipleura. In many insects, especially the more primitive groups such as Thysanura and Blatodia, the flagellum partly or entirely consists of a flexibly connected string of small ring-shaped annuli. The annuli are not true flagellomers, and in a given insect species the number of annuli generally is not as consistent as the number of flagellomers in most species. 10. In many beetles and in the chalcidoid wasps, the apical flagellomers form a club shape, called the clava. The collective term segments between the club and the antennal base is the funicle. Traditionally, in describing beetle anatomy, the term funicle refers to the segments between the club and the scape. Traditionally, in working on wasps, the funicle is taken to comprise the segments between the club and the pedicel. Quite commonly, the beyond the pedicel is quite complex in Endopterygota such as beetles, moths and Hymenoptera, and one common adaptation is the ability to fold the antenna in the middle, at the joint between the pedicel and the flagellum. This gives an effect like a knee bend, and such an antenna is said to be geniculate. Geniculate antennae are common in the Coleoptera and Hymenoptera. They are important for insects like ants that follow scent trails, for bees and wasps that need to sniff the flowers that they visit, and for beetles such as Scarabaeidae and Curculionidae that need to fold their antennae away when they self-protectively fold up all their limbs in defensive attitudes. Because the funicle is without intrinsic muscles, it generally must move as a unit, in spite of being articulated. However, some funicles are complex and very mobile. For example, the Scarabaeidae have lamellate antennae that can be folded tightly for safety or spread openly for detecting odors or pheromones. The insect manages such actions by changes in blood pressure, by which it exploits elasticity in walls and membranes in the funicles, which are in effect erectile. 12. In the group's more uniform antennae, for example, millipedes, all segments are called antennomers. Some groups have a simple or variously modified apical or subapical bristle called an arista. This may be especially well developed in various Diptera. 13. Functions. Olfactory receptors, scales and holes, on the antenna of the butterfly Aglaeo, electron micrograph. Olfactory receptors on the antennae bind to free-floating molecules, such as water vapor, and odors including pheromones. Neurons that possess these receptors signal this binding by sending action potentials down their axons to the antennal lobe in the brain. From there, neurons in the antennal lobes connect to mushroom bodies that identify the odor. The sum of the electrical potentials of the antennae to a given odor can be measured using an electroantennogram. 14. In the monarch butterfly, antennae are necessary for proper time-compensated solar compass orientation during migration. Antennal clocks exist in monarchs, and they are likely to provide the primary timing mechanism for sun compass orientation. 15. In the African cotton leafworm, antennae have an important function in signaling courtship. Specifically, antennae are required for males to answer the female mating call. Females do not require antennae for mating. A mating that resulted from a female without antennae was abnormal. 16. In the diamondback moth, antennae serve to gather information about a host plant's taste and odor. After the desired taste and odor has been identified, the female moth will deposit her eggs onto the plant. 17. Giant swallowtail butterflies also rely on antenna sensitivity to volatile compounds to identify host plants. It's found that females are actually more responsive with their antenna sensing, most likely because they are responsible for oviposition on the correct plant. 18. In the crepuscular hawk moth, Manduca sexta, antennae aid in flight stabilization. Similar to halteres and dipteran insects, the antennae transmit Coriolis forces through the Johnston's organ that can then be used for corrective behavior. Flight stability studies in which moths with flagella amputated near the pedicel showed significantly decreased flight stability over those with intact antennae. 19. To determine whether there may be other antennal sensory inputs, a second group of moths had their antennae amputated and then reattached, before being tested in the same stability study. They showed slightly decreased performance from intact moths, indicating there are possibly other sensory inputs used in flight stabilization. Re-amputation of the antennae caused a drastic decrease in flight stability to match that of the first amputated group. Structure. Electron micrograph of antenna surface detail of a wasp, Vespula vulgaris. The three segments of the typical insect antenna are the scape or scapus base, the pedicel or pedicellus stem, and finally the flagellum, which often comprises many units known as flagellomers. The pedicel, the second segment, contains the Johnston's organ, which is a collection of sensory cells. 11. Scape is mounted in a socket in a more or less ring-shaped sclerotized region called the torulus, often a raised portion of the insect's head capsule. The socket is closed off by the membrane into which the base of the scape is set. However, the antenna does not hang free on the membrane, but pivots on a rigidly sprung projection from the rim of the torulus. That projection on which the antenna pivots is called the antennifer. The whole structure enables the insect to move the antenna as a whole by applying internal muscles connected to the scape. The flexibly connected to the distal end of the scape, and its movements in turn can be controlled by muscular connections between the scape and pedicel. The number of flagellomers can vary greatly between insect species, and often is of diagnostic importance. Flagellomers are connected by membranous linkage that permits movement, though the flagellum of true insects does not have any intrinsic muscles. Some other arthropoda do, however, have intrinsic muscles throughout the flagellum. Such groups include the Symphyla, Collembola and Dipleura. Many insects, especially the more primitive groups such as Thysanura and Blatodia, the flagellum partly or entirely consists of a flexibly connected string of small ring-shaped annuli. The annuli are not true flagellomers, and in a given insect species the number of annuli generally is not as consistent as the number of flagellomers in most species. 10. In beetles and in the chalcidoid wasps, the apical flagellomers form a club shape, called the clava. The collector of the segments between the club and the antennal base is the funicle. Traditionally, in describing beetle anatomy, the term funicle refers to the segments between the club and the scape. However, traditionally in working on wasps, the funicle is taken to comprise the segments between the club and the pedicel. 10. The funicle beyond the pedicel is quite complex in Endopterygota such as beetles, moths and Hymenoptera, and one common adaptation is the ability to fold the antenna in the middle, at the joint between the pedicel and the flagellum. This gives an effect like a knee bend, and such an antenna is said to be geniculate. Geniculate antennae are common in the Coleoptera and Hymenoptera. They are for insects like ants that follow scent trails, for bees and wasps that need to sniff the flowers that they visit, and for beetles such as Scarabaeidae and Curculionidae that need to fold their antennae away when they self-protectively fold up all their limbs in defensive attitudes. Because the funicle is without intrinsic muscles, it generally must move as a unit, in spite of being articulated. However, some funicles are complex and very mobile. For example, the Scarabaeidae have lamellate antennae that can be folded tightly for safety or spread openly for detecting odors or pheromones. The insect manages such actions by changes in blood pressure, by which it exploits elasticity in walls and membranes.